You are welcome once again to Bio 211, the Plant Physiology Unit 1, Session 5, Light Dependent Phase of Photosynthesis. And by the end of this session, students should be able to state the roles played by ATP, NADPH in carbon dioxide reduction, list three phases of the carbon cycle, show the relationship between light dependent and light independent phases of photosynthesis, give account of carbon dioxide fixation, carbon dioxide reduction, Rubisco or ribulose biphosphate regeneration, outline plant adaptations to environmental conditions in carbon dioxide fixation, and state the products of light independent phase of photosynthesis. The light independent phase, otherwise called light independent reactions, are the second step of photosynthesis. The reactions are light independent because light is not directly required for the reactions to proceed. However, the ATP and NADPH produced during the light phase are used to reduce carbon dioxide. So this is the carbon cycle, and you have the chloroplast. Within the chloroplast you have the stroma, you have water coming in through the thylakoid membrane, that's the granum, the outer membrane, inner membrane, ADP plus pi, that's a phosphate, giving us NADP, and NADP, it goes through the intergranum, then gives us ATP plus NADPH. Then we move on to the cycle where carbon dioxide is introduced, and we have GA3P. The reduction of carbon dioxide occurs in the stroma of the chloroplast by means of a series of reactions known as the carbon cycle. So the figure is meant to give you a relational impression of where sugar manufacture takes place inside the chloroplast. So this is the chloroplast with its various components: the inner membrane, the outer membrane, the granum, the lumen, the stroma lemli, the thylakoid membrane space. You have the stroma and others. So an overview of the Calvin cycle. The process is named after Melvin Calvin and his associate who discovered the process. The Calvin cycle includes carbon dioxide fixation, carbon dioxide reduction, and the regeneration of ribulose biphosphate, also known as Rubisco. So here we go. We have three molecules of carbon dioxide. That's the Rubisco stage 1. We have carbon dioxide fixation. We have 6 molecules moving in through a cycle. You have 6 ATP giving us ADP. It is reduced. Then stage 2, that's the reduction, and NADPH is reduced to NADP and plus. Then it moves on. We have 6 molecules. It goes through another process, and we have 1 molecule and half of the molecule of glucose. Then it moves. Then it reduces to 5 molecules. Regeneration of the ribulose biphosphate, that's the third stage. Then you have 3 ADP coming out from the 3 ATP. So we have 3 molecules at the end of the day. So that is what transpires during the Calvin cycle. So an overview of the Calvin cycle is that ATP and NADPH produced by the light reactions are used in the Calvin cycle to reduce carbon dioxide to sugar. The Calvin cycle is similar to the Krebs cycle in that the starting material is regenerated by the end of the cycle. Carbon enters the Calvin cycle as carbon dioxide and leaves as sugar. So ATP is the energy source, while NADPH is the reducing agent that adds high energy electrons to form sugar. The Calvin cycle actually produces a 3-carbon sugar glyceraldehyde 3-phosphate G3P. For the Calvin cycle to synthesize 1 molecule of sugar, that's glyceraldehyde 3-phosphate, 3 molecules of carbon dioxide must enter the cycle. Let us turn now to the 3 phases. So now what is carbon dioxide fixation? It is the attachment of carbon dioxide to an organic compound. Carbon dioxide fixation is the first event in the Calvin cycle. Carbon dioxide fixation takes place when carbon dioxide combines with a 5-carbon compound called ribulose biphosphate. An enzyme called ribulose biphosphate carboxylase, otherwise known as ribisco, speeds up the reaction. This enzyme is one of the most abundant proteins on Earth. When carbon dioxide combines with ribulose biphosphate, a 6-carbon molecule is formed, which immediately splits up to yield 2 copies of the same 3-carbon molecule called phosphoglyceric acid, PGA. This reaction proceeds immediately after the 6-carbon molecule formed during carbon dioxide fixation splits to yield the carbon and the 2 PGA. This is an endogenic reduction phase. It is a 2-step process that couples ATP hydrolysis with the reduction of 3-phosphoglycerate to glyceraldehyde phosphate. An enzyme phosphorylate 3-phosphoglycerate speeds up the process by transferring a phosphate group from ATP. The reaction A produces 1,3-biphosphoglycerate. B uses 6 ATP molecules to produce 6 molecules of 1,3-biphosphoglycerate. C primes 1,3-biphosphoglycerate for the addition of high energy electrons from NADPH. 2. Electrons from NADPH reduce the carboxyl group of 1,3-bisphoglycerate to the aldehyde group of glyceraldehyde 3-phosphate. The product, glyceraldehyde 3-phosphate, stores more potential energy than the initial reactant 3-phosphoglycerate. Glycerate 3-phosphate is the same 3-carbon sugar produced when glycolysis splits glucose. So for every 3 carbon dioxide produced that enter the carbon cycle, 6 3-carbon molecules are produced, only one of which can be counted as net gain. The cycle begins with 3 5-carbon ribulose biphosphate molecules, a total of 15 carbons. The 6 3-glycerate 3-phosphate molecules produced contain 18 carbons, a net gain of 3 carbons from carbon dioxide. One G3P molecule exits the cycle. The other 5 are recycled to regenerate 3 molecules of ribulose biphosphate. So in carbon dioxide reduction, each PGA is reduced by 1 NADPH and ATP formed in the thylakoid membrane during the light-dependent reactions. The resultant product is called glyceraldehyde 3-phosphate or phosphoglyceraldehyde. PGAL. PGAL is the end product of the Calvin cycle. 2 PGAL molecules then combine to form 1 molecule of glucose, which then becomes the end product of photosynthesis. So in here you have all the steps that one is supposed to go through, as we have discussed, from step 1, carbon dioxide fixation, where you have 12 ATPs being reduced to 12 ADPs. Then we have 12 NADPH, 12 NADP plus, that's step 2. Then we have 12 3-GDP, 12 G3P, moving into the next step, which is step 3, and we have ribisco. That's, you have 2 molecules of G3P and giving us 6 molecules of ATP. That's ADP being produced, and that is step 4, with which we have 6 ribulose 1-biphosphate being produced. Then the cycle continues on a bit. Carbon dioxide reduction. So notice that the carbon cycle is actually made of the carbon dioxide fixation and reduction. Try out a step-by-step outline of the cycle in your jotter. You get this. Ribulose biphosphate combines with carbon dioxide to form 6-carbon molecule. The 6-carbon molecule immediately breaks down to 2 PGA, that's phosphoglycerate or phosphoglyceric acid. The phosphoglyceric acid is then reduced to phosphoglyceraldehyde, which is the end product of the carbon cycle. Outside the cycle, 2 phosphoglyceraldehyde molecules combine to give glucose 6-phosphate, a molecule that can be metabolized to other organic molecules. So in the cycle, 5 out of every 6 PGAL are used to reform RUBP, that's ribulose biphosphate. What happens to RUBP after it takes up carbon dioxide in carbon dioxide fixation? The statement simply means that RUBP is not used up. It's somehow remade for use again. How does this happen? Study carefully again figures and follow through on the step-by-step process of regenerating RUBP. A complex series of reactions rearranges the carbon skeletons of 5 G3P molecules into 3 RUBP molecules. These reactions require 3 ATP molecules. RUBP is thus regenerated to begin the cycle again for the net synthesis of 1G3P glyceraldehyde 3-phosphate molecule. The Calvin cycle uses the product of the light reactions, that's 9 ATP molecules and 6 NADPH molecules. So 3 G3P molecule produced by the Calvin cycle is the raw material used to synthesize glucose and other carbohydrates. The Calvin cycle uses 18 ATP and 12 NADPH molecules to produce 1 glucose molecule. Altogether, the ATP and the NADPH consumed in producing glucose represent about a 30% conversion rate. That is to say that 30% of the energy available in ATP and NADPH is finally stored in glucose. The Calvin cycle of carbon dioxide fixation and reduction are true of all plants, but in areas like the tropics and arid desert where temperatures can be very high with hot, dry air, plants use other pathways in addition to the Calvin cycle to fix and reduce carbon dioxide. We shall now turn our attention to these adaptations and the conditions that produce such responses or adaptations in plants. So, photorespiration. When the oxygen concentration in the leaves air spaces is higher than carbon dioxide concentration, rubisco accepts oxygen and transfers it to RUBP. This pathway produces no ATP molecules and reduces the number of organic molecules that can be reduced by the Calvin cycle. This metabolic pathway is called photorespiration and it reduces photosynthetic output. Photorespiration is a metabolic pathway that consumes oxygen, evolves carbon dioxide, produces no ATP and decreases photosynthetic output. It occurs because the active site of rubisco can accept oxygen as well as carbon dioxide. The respiration in photorespiration refers to the fact that this process uses oxygen and releases carbon dioxide. Whether photorespiration is beneficial to plants is not known. It is known that some crop plants, example soya beans, lose as much as 50% of the carbon fixed by the Calvin cycle to photorespiration. If photorespiration could be reduced in some agricultural plants, crop yields and food supplies would increase. Photorespiration is fostered by hot, dry, bright days. Under these conditions, plants close their stomata to prevent dehydration by reducing water loss from the leaf. Photosynthesis then depletes available carbon dioxide and increases oxygen within the leaf air spaces. This condition favors photorespiration. So photosynthesis and the resultant production of sugars occur during the daytime, but when climatic conditions force the plant to photorespire, available food is used up, and so food yield reduces to defeat the objective of food production. How do plants achieve their objective for photosynthesis in hot, arid environments? Many plant species precede the Calvin cycle with reactions that incorporate carbon dioxide into 4 carbon compounds. These plants are called C4 plants. The C4 pathway is used by several thousand species, at least 19 families including corn and sugarcane, important agricultural grasses. This pathway is adaptive because it enhances carbon fixation under conditions that favor photorespiration, such as hot, arid environment. Leaf anatomy of C4 plants spatially segregates the Calvin cycle from the initial incorporation of carbon dioxide into organic compounds. In C4 plants, there are 2 distinct types of photosynthetic cells. Bundle sheath cells are arranged into tightly packed sheets around the veins of the leaf. Thylakoids in the chloroplast of bundle sheath cells are not stacked into grana. The Calvin cycle is confined to the chloroplast of the bundle sheath. Mesophyll cells are more closely arranged in the area between the bundle sheath and the leaf surface. The Calvin cycle of C4 plants is preceded by incorporation of carbon dioxide into organic compounds in the mesophyll. The steps of carbon fixation. Step 1. Carbon dioxide is added to phosphoenol pyruvate to form oxaloacetate, a 4-carbon product. PEP, phosphoenol pyruvate carboxylate, is the enzyme that adds carbon dioxide to PEP compared to Rubisco. It has a much greater affinity for carbon dioxide and has no affinity for oxygen. Thus, PEP carboxylates can fix carbon dioxide efficiently when Rubisco cannot under hot, dry conditions that cause stomata to close, carbon dioxide concentrations to drop, and oxygen concentrations to rise. Step 2. After carbon dioxide has been fixed by mesophyll cells, they convert oxaloacetate to another 4-carbon compound, usually malate. Step 3. Mesophyll cells then export the 4-carbon product, that is malate, to the bundle sheath cells. In the bundle sheath cells, the 4-carbon compounds release carbon dioxide, which is then fixed by Rubisco in the Calvin cycle. Mesophyll cells thus pumps carbon dioxide into bundle sheath cells, minimizing photorespiration and enhancing sugar production by maintaining a carbon dioxide concentration sufficient for Rubisco to accept carbon dioxide rather than oxygen. A second photosynthetic adaptation exists in succulent plants adapted to very arid conditions. These plants open their stomata primarily at night and close them during the day, opposite of most plants. This conserves water during the day but prevents carbon dioxide from entering the leaves. When stomata are open at night, carbon dioxide is taken up and incorporated into a variety of organic acids. This mode of carbon fixation is called crassulacean acid metabolism, CAM. The organic acids made at night are stored in vacuoles of mesophyll cells until morning, when the stomata close. During daytime, light reactions supply ATP and NADPH for the Calvin cycle. At this time, carbon dioxide is released from the organic acids made the previous night and is incorporated into sugar in the chloroplast. What differences and or similarities do you notice between the two pathways? Write down your observations before you read on. The two pathways are similar in that carbon dioxide is first incorporated into organic intermediates before it enters the Calvin cycle, but the two differ in that the initial steps of carbon fixation in C4 plants are structurally separate from the Calvin cycle. In CAM plants, the two steps occur at separate times. When you go back to the simplified diagram of the Calvin cycle, you notice that the main product of photosynthesis is glucose, B-phosphate. What would you also notice that from glucose, the following products are obtained: sucrose, glucose, starch, which is the form in which excess carbohydrate is stored in plants. Glucose may be used by the plants to provide energy for its use. Carbon dioxide and water result from the breakdown of glucose. Glucose may also combine with elements such as nitrogen and sulfur to produce proteins. Lipids for cell protoplasm are produced from glucose, and finally, but not the least, cellulose, the material that makes up the structure of cell walls, is also a polymer of glucose. Oxygen is a byproduct of photosynthesis. The plant uses part of the oxygen it produces in its cellular respiration, but most of it is released to the atmosphere. So in review, the light independent phase of photosynthesis uses a pathway that is cyclic and called the Calvin cycle. It is a 3-phase process involving a C3 intermediate. However, in hot and arid environments, plants photorespire or produce carbon dioxide from a C4 intermediate to feed the Calvin cycle. The products of the light-independent reactions of photosynthesis include sucrose, glucose, starch, proteins, lipids, and cellulose. Oxygen is released as a byproduct.